Thanks for joining. Hi, everyone. Thanks for joining the first BIRCH Methods Core invited seminar given by Professor Ying Qing Chen at Stanford Prevention Research Center and the Director of Palo Alto Veteran Affairs Cooperative Studies Coordinating Center. Professor Chen has made major contributions in the development, design, management, and analysis of clinical trials and observational studies with a focus on population-based disease prevention. Among other studies he participated in, he led the Statistical Data Management Center for the HPTN 052 study, recognized as the scientific breakthrough of the year 2011 by the journal Science. Professor Chen is also recognized by being fellow of the American Statistical Association. Without further ado, let's welcome our speaker to get started.
Hey. Thank you, Gary. I'm really happy to be back to talk to folks mostly in Seattle based. So I saw our old friends and colleagues also in the seminar, so I won't say hi. I won't say everyone's name, but please, it's great to see you all. So I would like to thank the center, especially Brian and Angela, for helping me put up the slides and get things going. And so it is great. The topic I'm going to talk today is surrogate markers and how to measure surrogacies in clinical research. And the data example, the study example I used is landmark HIV clinical trials for treatment as prevention. So yeah. So this study for those of you who are working in HIV clinical trials probably know this trial very well. It's to compare long-term effectiveness of two ART strategies in HIV transmission among serodiscordant couples. So it is it's to study at that time if immediate therapy to provide HIV, to provide antiretroviral drugs to HIV infected patients when they are yet to develop serious symptoms, and if early therapy or immediate therapy can help prevent HIV transmission from HIV infected partners to their loved ones who are HIV seronegative at that moment. So the study populations is HIV discordant couples with index partners of CD4 counts between 350 to 550. And when the study was first designed, it's actually the CD4 count was between 300 to 500. But later on, after we consulted with the DSMB and consulted with experts in the area, and we decided to raise the CD4 counts by 50. So the enrollment CD4 counts were between 350 to 550. And there are two treatment strategies. One is the immediate therapy. So it's the ART initiation upon enrollment. So which means as soon as the index partners got screened and enrolled, they were starting ART at the enrollment. And the comparative treatment strategy is so-called delayed therapy. So it's the ART initiation when the index partner CD4 counts falls between 200 to 250 or developing AIDS-defining elements. So essentially, that was the treatment guidelines, especially in developing countries. not necessarily in the U.S. though, but that was the de facto standard of care at that time. So this trial was designed to be multi-site, two-arm, control randomized trial, 1-to-1 into treatment versus control in 3 continents, 8 countries, 12 clinical sites. It's actually 4 continents, 9 countries, and 13 clinical sites if we include 1 site in Boston in the US. And we intended to this trial to have 1.5-year accrual with 5 years of follow-up. And so each participant was supposed to follow up for 5 years, so up to 6.5 years of total study durations. But in fact, the accrual was not as fast as we designed it to be. It actually took about 36 months for the accrual to complete. So the total was close to 10 or 11 years if we count the study running period with about 80 some participants enrolled for the running period. So the primary endpoint for this study is the incident HIV infection occurred in the partners of randomized HIV infected index cases. And we only count supposedly in the primary analysis, only a position from the index partners will be included. So each endpoint will need to be confirmed such that the viral envelope sequence in the index case matches that of the partners. So we want to make sure that if an infection happened, we can confirm that infection is actually linked between the HIV positive partner and negative partner who later got later become seropositive. So the study population of this trial is mainly in Africa, with 954 couples recruited from Africa. In Asia, we have 531 couples. In America, so we have 278 couples. But we, if I remember right, there was only 1 couple from the US side. So here's the interim results in 2011. Now it's almost over 10 years ago. So the results were very striking, and if we were looking at linked transmissions in the immediate arm, we had 1 transmissions in the delayed arm. We have 27 transmissions observed. So the hazard ratio is estimated to be 0.037. The efficacy is measured. Essentially, efficacy is a 1 minus hazard ratio, so 96%. So that the 96% number was cited widely about this the immediate treatment strategy efficacy. And we actually also look at all transmissions, regardless the transmissions being linked or not. And we observed 4 transmissions in the immediate arm, 35 transmissions in the delayed arm. So the hazard ratio estimated to be 0.114. The efficacy was calculated to be 88.6%. So we also look at the clinical events, so which is the we treat the each partner, each serodiscordant couple as a 1 tri-unit. So the HIV positive participant may experience some clinical events, plus the transmission event that would happen to the HIV negative partner. So when we look at the clinical events, defined with some criteria, we observed 40 events in the immediate arm, 65 from the delayed arm, so the hazard ratio to be estimated as 0.594. efficacy is estimated to be 40%. So this basically says means one index partner of the immediate arm taking drugs is not only going to benefit to the HIV negative participants in terms of prevention endpoint, but also it is beneficial to the HIV infected partners in terms of therapeutic endpoint. So it's and when we also look at the so-called composite events. So those are the serious clinical events that would happen to HIV positive partner and the transmission event. So we observe 23 events from immediate arm, from the immediate arm, 79 from the delayed arm, and the hazard ratio to be 0.265, efficacy estimated to be 73.5%. So if we are looking at the serious clinical events or composite events, we actually use those composite events to monitor this trial and to decide when the interim analysis to be done. And this is the one we observe about 85 events. We actually performed this interim analysis, and the trials results were quickly disclosed to the public and to declare the benefit of immediate therapies. But since this trial was designed to be a trial to compare immediate therapy versus the delayed therapies, by the time when we performed our interim analysis and had this striking results, and the contrast between the two arms were already there. So we decided to cross in the treatment, which means the delayed arm will all receiving therapies immediately, and to still let the trial run because the contrast is there. And so the final results were disclosed in 2015. So after May 11th, or after the first interim analysis, so we observed HIV infected events in the delayed arm to be 15, and in the early arm to be 15, in the delayed arm to be 17. So it's comparable because those events are accumulated already happened before. Most of those events happened probably during the crossing period. And so if you look at the linked 2 versus 7, and so the overall for the prevention endpoint, the linked events, the rate ratio between the early arm and delayed arm is 3 versus 43. The hazard ratio to be 0.07, and if you take a 1 minus the hazard ratio, that's the efficacy still remains to be 93%. So not much far away from 96% we observed when we performed the interim analysis. The overall HIV infection, regardless of linked or not, is 69%. So it is still, very striking in terms of risk reductions for immediate therapies, and looks like it is durable to somewhat degree in the trial settings. So this is the trial, and for clinical trials such as HPDN-052, it's a long-term clinical trials. Apparently, we have to monitor these trials, and we use the composite endpoints we just mentioned to monitor this trial, and developing monitoring plans. So the the composite endpoints is a combination of a severe event in HIV positive partners and plus viral transmissions would happen to HIV partners. So this is a schematic diagram what an event may happen to a couple in the two arms. For a trial like this, and we're lucky to design this trial to be relatively long-term and to have the resources. But for lots of long-term trials, especially when we are dealing with hard, long-term prevention endpoint, and it is somewhat desirable to find out if there is surrogate endpoints we can use or we can look at before to avoid collect endpoints which need a lot more resources and time to collect. So what kind of a surrogate endpoint we should look at for a trial like this in HIV settings? I know we have a lot of discussions and in literature, in practice, what should we do? And the interesting thing is, even though there are some agreement, but how do we assess or compare surrogate endpoint? It's still an issue. So a qualitative evaluation of a surrogate endpoint almost always, trace back to Ross Prentice's so-called Prentice criteria. So if we are using this notation, Z as intervention, S as a surrogate marker, T as a clinical endpoint. So conceptually, Ross proposed this Prentice criteria in 1989 Stats in Medicine paper. So basically, testing of the distribution of surrogate markers in the different intervention arms is equivalent to test the distributions of clinical endpoints in the different treatment arms. So, and operationally, we can look at the distributions of clinical endpoints given surrogate markers, and intervention should be equal to the distribution of clinical endpoints given surrogate endpoint, and the distribution of clinical endpoints in the surrogate markers is not the same as the distribution of clinical endpoints itself. So, and ever since the Prentice criteria were proposed, people have been thinking, oh, this criteria it's quite stringent. So in terms of biomarker surrogacy, if a marker is a perfect surrogate marker, the so-called perfect surrogate marker, if it is captures all the dependence of T clinical endpoints on the intervention in the sense of the conditional independence of clinical endpoints with regard to the treatment assignment, but the distribution of T of the clinical endpoints given an intervention will still depend on the markers. So this is a condition. This condition of perfect surrogacy is that's equivalent to Prentice criteria, and the opposite of this criteria, and oftentimes we call it the surrogate marker, is useless. So if it does not account for any dependence of T on Z in the sense of the conditional independence or the marker as it's independent of the treatment. But there is a wide range between these two extreme scenarios, either completely useless or perfect surrogate markers. It's kind of interesting, and there are a lot of scenarios. A marker we identified, especially if we're looking at immunological biomarkers, and those markers can be partial surrogate markers if it captures some of the treatment effect on the clinical endpoint, but not all of that, especially when the clinical endpoint is a composite endpoint. It's not necessarily the one the transmission happened. So like the composite endpoint we observed, we used. So a marker is neither a perfect surrogate marker nor a completely useless surrogate marker. So how do we assess the partial surrogacy, or if we have a couple of them, or if we have many of those partial surrogate markers, how do we rank those surrogate markers? It can be interesting to practice. So let's get back to qualitative evaluation using Prentice criteria. So operationally, if we are looking at the distribution of clinical endpoints given surrogate markers and intervention, if it is equal to the distribution of clinical endpoints given surrogate markers, essentially this is somewhat adjusted outcomes by S, right? So so basically we stratify the surrogate marker distributions, and then we look at the conditional distribution of clinical endpoints in the interventions in that stratum, and if we can evaluate if they're equal or not. So this kind of adjusted idea prompts people to think about how to measure surrogacies of surrogate markers. And one leading one is the so-called PTE. It's the proportion of treatment effect explained. For example, in the binary outcomes, people would use two logistical models, logistical regression models. So one with intervention only, the other one with intervention plus surrogate markers. And then, to fit the two models and get the regression parameter estimates, say beta in the model with intervention only, or beta S and phi Z in the model with both intervention and surrogate markers, and then take the difference between the two regression parameter estimates and divided by the beta, the regression parameter estimate from the intervention only model. And then that's called, it's kind of a difference, percentage of difference with respect to the regression parameter estimated from the intervention only logistic regression models. So basically, it is to compare unadjusted and adjusted model by surrogate marker distributions, regardless the underlying biological mechanism in subgroups. So this kind of approach essentially measures population impact associated with a candidate surrogate markers. So if S is the represents the surrogate markers. So it's a more of an empirical association type of way of assessing the surrogacy of surrogate markers as, because regardless of what kind of mechanism it is, or regardless what kind of pathway it is. So the issues on PTE is for perfect markers, PTE is 1, that's good. For useless markers, the PTE is 0, that's good. But the problem is the value of PTE alone, however, cannot determine the surrogacy levels of candidate biomarkers, because totally regardless disregard the mechanism. The PTE close to 1 implies that good surrogate markers only if it is known that the intervention effect is primarily mediated by a marker. But a PTE close to 1 may also happen when the intervention has harmful or toxic effects that are not mediated via the biomarker. So PTE is not necessarily restricted to unto 0 and 1 range. So if in the adjusted treatment, the regression parameter changes its direction, then the PTE is then greater than 1 without some understanding of actual biological mechanism. The interpretation of PTE can be quite challenging. But nonetheless, PTE offers a measure of the percentage of differences of regression parameters associated with the biomarker. So the other thing, and people often criticize about PTE is that PTE is model dependent, which means it really depends on the two logistic regression models. And the issue is we don't know if those two models are correct or not, or if one of them is correct, or both of them are correct. So both models are actually serving as working models, and also in terms of the actual variance of PTE, estimated PTE, can be quite large because you have a regression parameter estimate in the denominator, and so that can lead to quite large variance. So unless we have a big sample size and then the PTE's variance oftentimes is too big and to lead us to any more confirmative hypothesis testing. So so those are the issues. And then
hey, Ying.
Yes.
Sorry, I have a quick question. So what do you mean by like the PTE may be close to 1 if the intervention have some toxic effect? Is it something not captured by T? So your next slide. So like when talking about the range of PTE, so PTE when pretty close to 1, it may also happen when the intervention has harmful or toxic effect, not mediated via the marker. So does it mean like the intervention has some other effect that's not captured by our primary outcome T?
Yes.
Okay.
So that's basically what this means. So if you look at it's just missed that part. So
great, thank you.
Mm-hmm. Okay, so those are the issues. Thanks for the questions, Faye. So a second approach, so people are trying to still use adjusted approach, but using a model-free kind of approach. So not necessarily rely on the two logistic regression models people tend to use in the PTE to calculate the PTE. So this is a measure is called the F measure, proposed by Jeremy Taylor and one of his students in a 2002 biometrics paper. So this F measure is also actually equivalent to a percentage or proportion of a treatment effect. And in their proposal, they look at the amount, to look at the difference of the clinical endpoints in the two intervention arms, and it's actually a total effect, right? And the treatment effect on the additive scales, the difference of these two distributions, and then to look at what if we will be using instead of what if we are going to apply the conditional distributions of clinical endpoints given a surrogate marker in the intervention arm, but to the surrogate marker distributions in the control arm. So we have the absolute risk from the control arm, but can we use the absolute risk, conditional absolute risk given a surrogate marker, but using the control arms surrogate marker distributions as a reference population, and recalculate the incidence that would expect to be to the control arm, then take that difference, then we can see how that differences would compare with each other, or the proportion of that expected difference versus the overall treatment effect, and they call it adjusted treatment effect through some kind of a direct adjustment, and then take one subtract of that. And so, or more generally, they proposed this AABB and AB. So essentially, it is a function of some kind of adjusted absolute risk, and then take the so-called F measure to be AA subtract AB versus AA minus BB. So if you look at the AA, it's basically the absolute risks given a marker over one arm, arm A, and the surrogate marker distributions in arm A, and BB is the conditional distributions given a marker given in the arm B group, and apply to the arm B group's surrogate marker distributions. But AB is just like more like a cross over using A in one arm's distribution of clinical endpoints given surrogate markers, but apply to another arm's surrogate marker distributions. So this is a more general formulation of the F measure Wang and Taylor proposed. So if we look at that in this schematic plot, so let's say if we are looking at a control arm, what we observe is we basically stratify the control arm into two groups, into marker groups versus the groups without the marker, or the surrogate marker equals 1, or the other surrogate marker equals 0 for binary surrogate marker. And we have the two distributions, so we can use total probability to calculate the probability of, or the distribution of clinical events in the control arm. But a hypothetical group or expected group is same kind of stratification of the markers, but instead of using the actual marker we're going to see in that control arm, but we apply the conditional distributions of clinical endpoints given the marker, but from the intervention arm to the hypothetical to the structure of markers in the control arm. So this kind of indirect adjustment for marker distributions. So in the alternative form for F measure, for the binary marker, we can rewrite it to be a probability of P clinical endpoint we observe in the intervention arm, subtract a P star. So this is the expected distribution of clinical endpoints given Z equals 1. It's actually and over probability of the T equals 1 given Z equals 1. So this is the distribution of clinical endpoints in the intervention arm, subtract. This is the total effect we observed. So we can see that cross-in or cross product to be an expected event with control arm surrogate marker structure. So we apply that. So that's the differences, and the ratio of these two differences is actually the F measure. So F measure, as it is formulated, it's model free. It does not rely on any models, and but it still does not solve all the PTE issues. The strong assumption of biological mechanisms are still necessary for it to be bounded onto 0, 1. When there's again, same issues when there's unexpected reverse treatment effect, given some surrogate values, the F measure may lie out 0 and 1. The variance of the estimated F measure can also can be large. Superficial solution is transforming the F measure by this transformation, so 1 over 1 plus 1 over F minus 1 squared, and it's kind of there's actually a special term for that. It's called the PCS. It stands for proportion of treatment effect captured by candidate surrogate marker endpoints, coined by Fumiyaki and Kuruki in 2014 Stats Medicine paper. So there are issues too, with the transformed F measure. So different situations may lead to same PCS values. So it is challenging to use PCS to rank candidate surrogate markers as well, and especially when you have this square term. So those are the.
I'm sorry, Ying, I just want to let you know it's 10 after 11. You asked me to keep a time.
Okay, great, thank you. Right. So in terms of the suppressing, the HPD-052 examples, we know suppressing viral load is how ART controls AIDS-related events progression and potentially HIV transmissions. And so essentially, there is a strong biological mechanism we believe, or we have evidence, antiretroviral therapies will work on viral load, and the viral load, lower viral load will lead to slower disease progression and lower risk of HIV transmission. So that's the good news to apply F measure onto HPD-052 studies, because we do have this kind of strong biological mechanisms. So we look at the viral load greater than 1K copies at year 1 as surrogate markers, and we can assess, for example, survival status by year 3. So we can also look at the viral loads at year 1 survival status 3, and with the severe endpoint in HIV positive but events in HIV positive partner and HIV transmission event happens to HIV negative one. So we can extend F measures to even time varying, not just binary outcomes. And so we pick up a specific time and look at beyond that time, see and what the surrogate marker would look like. So this T can be a landmark time, and the C is actually the survival time. So we can use the denominator, the total effect in the risk set, and then we adjusted to calculate differences by adjusted treatment effect in the risk set. So this is a schematic plot of that time varying F measure for useless to partial markers. So the flat lines, the useless biomarkers would be represented by that time varying functions, and for different partial markers. And this on the right is the perfect two partial markers. The perfect markers is the solid lines, and the partial markers, how the partial markers may compare to that perfect markers. So we can certainly estimate and make inferences for categorical biomarkers. So we can assume random censoring and survival probabilities can be estimated by Kaplan-Meier estimates. The market distribution can be estimated by empirical distributions. And then we worked out all the technical details to calculate the asymptotics for this time varying measures. So we did run some simulations using Cox-Weibull distributions, basic Cox model with a Weibull underlying baseline distribution, and we run on 20,000 subjects, so 1,000 replicate simulations. So when you look at the coverage probabilities, it's works pretty well at for different scenarios. So for the F measure for the HPTN-052 studies, so we so this is a table to show at a different landmark time, year 2, 3, 4, 5, 6, 7, and in the delayed arm, what is going on in the delayed arm, what's going on in the immediate arm in terms of the prevalence of viral loads more than 1,000 copies among HIV-infected partners, and how the F measure would be calculated. So for year 2, it's 0.18, year 3, 0.41, year 4 is 0.52, year 5 is 0.72, year 6, 1.12, and year 7 is 0.812. And it looks like the surrogacy is, as time progresses and the viral copies more than 1,000 among HIV-infected partners, getting better, close to the clinical endpoints, as you can imagine, right? So it's not surprising, and still, because the get to the end of the study, so we have less number of events, clinical events to be calculated. So the range of the confidence will becomes big, and also we have this auto range value of even more than 1. So so that is the one extension we did using F measure, but extended to the time varying scenario. So with multiple landmark time. And then the other thing is we want to understand what is really F measure really doing. So if we can recall F measure, the numerator basically is the difference of what we observed and what we expected, using control arm as the reference group. So then it would be actually the same as the numerator of the so-called population attributable fractions if the treatment assignment were considered as an exposure of interest in the epidemiological investigation. So this is the population proportion of attributable fractions. If you look at the numerators, they are almost identical. The only difference is the denominator. In the population attributable fractions, it's just you look at the absolute risk of a disease, regardless of exposure, right? But in the F measure, you basically take the denominator as the difference of the total effect. So in fact, the F measure is equivalent to attributable fractions by a factor. And this factor is the ratio of percent of a reduction of immediate endpoint exposure by treatment arm, and the RD is another measure. So then we move on to say, can we just look at the population attributable fractions counterpart directly? Look at the so-called row measure. So basically, we take the difference of the observed and expected, but over the absolute risk in the intervention arm. So then it just becomes just the attributable fractions. So it provides an empirical absolute information of the moderated risk associated with the different market distributions with a range of 0 to 1 for protective treatment effect. So now this row measure, unlike F measure, may go beyond 1, be greater than 1. This row measure, just like a proportion fraction, the proportion of attributable fractions is actually will always be between 0 and 1. So here is an example for binary surrogate markers in the 2-arm trial. And we have control arm, treatment arm marker distributions for clinical endpoints, we look at subtotals. So if we assume multinomial distributions, and then the row measure is just a differences as a denominator, as a numerator over the absolute risks in the intervention arm as a denominator, we can estimate it. We can estimate the confidence intervals. It's a long form, but it's doable. So we can actually use that relationship to assess what's the relationship between the PT pi measure or the F measure and this new row measure. And here are the graphs, some schematic graphs. So row measure is essentially it's just an F measure of the factor of 1 subtracted 1 over relative risk. So the row measure in the HPT-052 studies, so if we compare immediate versus delayed, and here are different markers we would compare. I know this table is pretty busy, but we did look at the different combinations. So instead, in addition to the viral load, we also add the CD4 counts as markers and the different combinations and different levels, and to look at how the different combinations to mimic what if we have different kind of candidate markers we would be looking at, and we can calculate their distributions in the delay arm, in the immediate arm, and then we can calculate the PTE, the F measure, and the row measure for different combinations. And it's not surprising to see F measure and row measure are pretty much equivalent. But only by a factor of 1 minus 1 of relative risks. So those are the results. I'm not going to go through each one of them. And basically, we did look at how do we rank them, and it looks like if you look at pick up a number, let's say at the year 2. Look at the viral load of less than 400 versus viral load of less than 1,000. The F measure gives to almost a perfect surrogacy in terms of surrogate. They're them as a surrogate markers, and the row measure also even on the different directions, but if you look at the magnitude, they're about the same. So in our paper, we actually also discussed the relationship between the row measure and the natural direct effect and the natural indirect effect under the counterfactual framework. There is a connection between these two. If, for example, in a confounding free scenarios, the numerator of the row measure actually captures the indirect effect to the marker immediate pathway. In that scenario, so the row measure is the proportional risk reduction that attributed to the marker mediated treatment effect, which entails a natural indirect effect in prediction. So in randomized clinical trials, and probably with a strong biological mechanism like in 052, we know the viral load directly related to the clinical endpoints and the transmission endpoint. So the row measure provides empirical sensible interpretation. So, for biomarker surrogacy assessment, there are other approaches as well. There are meta-analytic approaches, there are causal effect approaches. So there's a lot of literatures on that, and it's not limited to use a PTE or F measure. How come? So that's basically what we have done for the assessing surrogate markers for this important clinical studies. And so I would like to thank HPT-052 study participants and the protocol team, and our funders, Bates from NIH. And especially I would like to thank our UW-Hutch colleagues. And I have been working with UW-Hutch between 2004 and till last year. And many of my colleagues at Hutch and UW, we had a lot of discussions and conversations about on these issues. Many productive conversations with Ross, Tom, and Jim. I saw you were there, and Deborah Donnell, and Peter Gilbert, and certainly Gary and Chongji. And thanks so much for your conversation, for your help. So with that said, I probably can answer some of the questions if you have.
Thank you very much, Ying.
Mm-hmm.
I don't know how you clap in Zoom seminars, but maybe if people have questions, they could do the hand raise thing. Oh, Gary's doing it. Thank you. Or if someone just wants to pipe up with any questions.
I have a question, just to get us going. I was wondering, so because I'm just wondering if you could use this approach to look at the optimal viral load, for instance, Ying, that would be associated with the greatest impact on your outcomes. So instead of using arbitrary cut points like 1,000 or 400.
Yeah, that's a very good question. And one direct application certainly is that if we do have some meaningful, way or to do the cutoffs, so we can compare them, right? We can just calculate the F measure or the rho measure for each cutoff. Then we can run hypothesis testings because we already calculated the asymptotics. And the other thing is that, how do we figure out what is optimal based on that? We can certainly use this F measure or rho measure as objective functions and for a given cutoff. And then we can, optimize with respect to the cutoff. Oftentimes the thing is when we run that kind of optimization, it's more or less study dependent, depending on what kind of data we have. And can we use that optimal cutoff we got it from the study to another study? Sometimes it's hard to adjudicate, and so that's why in reality people just tend to use one cutoff. Let's say it's more or less like an age cutoff. You use 5 year, 3 year, and why 5 year, 3 year? There's no like a hard clear cut why we're doing so. So I think the other way of looking at it is probably to look at the different studies probably through a meta-analytic approach and to look at it. But then certainly the issue is there are different exclusion criteria, different quality, the amount of different studies, and how do we get that settled? And there are issues.
Yeah, I'd love to see a meta-analysis of some sort on that, just because it would have some practical implications in terms of counseling patients. If there was sort of a range or sort of an area you could say, well, in terms of your personal health, keeping your viral load below such and such a cut point looks like it might be, optimal. Whereas for HIV transmission risk, maybe that's higher or lower in terms of what they shoot for. And to some patients that might make a difference in how they think about suppression and adherence. Yeah.
And we didn't really talk about the adherence and compliance issues in this talk yet. If later time we have more time to interact, I can show you other work we did. How do we monitor viral load over time in terms of their compliance issues and how that would affect their clinical endpoints? And yeah, it's a tough issue though. So.
Any other questions? One thing I thought of while you were answering Susan's question, besides optimizing a viral load as a surrogate, you want to optimize the time window too. Like in a lot of mediation research and developmental research, the timing of measurement is often just an artifact of the research design and the funding and how often you can collect data, rather than focused on the unfolding process and what the time of all these different things are and when optimal moments of capturing associations are. And that's a hard thing to figure out.
Yeah, that's a very good question, Brian. Is in clinical trial settings, we somewhat have that kind of luxury, right? We ask patients to come into clinic every 4 weeks, every 6 weeks, or every 3 months or quarterly, or we have that kind of luxury. In the pure observational settings, not necessarily we can have that kind of luxury, and so the timing of doing that can be quite challenging. Yeah, and it is it's a surrogate marker has been there, and the statisticians have dedicated so much time and effort to trying to study it, via all kinds of approaches, or structural equation models, and to run joint modeling, and it has been quite sticky, and even today it's still hard. And in HIV prevention trials, and I believe the hard clinical endpoint of transmission is still the gold standard. People would like to do that. It's just and somewhat lucky. We know very well in the treatment as prevention settings, we know the viral load is the key issues. So but for in other settings, when the things are not so clear, especially for example in HIV vaccine studies, when you have different kind of immunological biomarkers, and we're not so sure which one really can which one is the clear winner for as a surrogate marker for HIV vaccines, then it is hard. So, but at least we want to rank those candidate markers. It's all about competing interests, right? So how do we rank them using a uniform or consistently defined measure? I think it's still useful, helpful.
Thanks.
I guess one other, I'm going to jump in since while other folks are thinking. One other thing that occurred to me is that this is somewhat, you basically are going from the intention to treat analysis to an approach that's sort of more analogous to per protocol, in which the intent to treat and the surrogate marker are more aligned with what actually happened in the trial. And so, there's some, I guess it's sort of like moving towards what may be most explanatory, but may not be achievable in the real world.
Yeah, that's a good way to look at it. So basically what we are doing here, or people are looking at here, is you see the difference between intervention arm and the control arm, right? And you just want to understand what this marker is the role in that difference you're saying. So basically, the F measure is saying, well, it's the same idea as in the PTE formulation, right? Is if we look at the intention to treat differences, and then we add in surrogate markers, and then we calculate the adjusted treatment effect, and then we can compare those two. So basically, that's the idea. So that's why the philosophy is the same. It's just provide an empirical measure, but the issue is it does not really incorporate what's really going on with the markers and the interventions and the clinical endpoints. So so I think to really trying to understand the causal relationship between intervention, marker, and clinical endpoints And I think definitely more detailed modeling are needed, regardless if it's more conventional pathway analysis or more counterfactuals, kind of put in the counterfactual settings to estimate the causal estimate, to propose a meaningful causal estimate and to estimate it, and so, or to even run or simulate clinical studies and to check different assumptions, and to look at to wiggle a bit and to find out what's really going on. And sometimes it's a bit, to me, it's a bit beyond the statistical scope. It requires additional knowledge to do that. But this measure, like you said, it's basically just to look at the overall difference you see, how much would be explained by surrogate markers in the association sense.
It strikes me as something, as we're the BIRCH Center and we're looking at mental health. Mental health is an outcome for which the causal pathways and the potential surrogate markers are much, much less clear, for instance, than HIV treatment. And, the idea that, this is somewhat like a mediation analysis, but, just the idea that we need to try and dig into the potential surrogate markers that might actually help people achieve mental health outcomes, whether biologic or scale-based measures. I think it's really an interesting area.
Yeah, I totally agree, especially for a lot of endpoints in mental health research. And it's not simple. I mean, even to look at an endpoint, it's not simple. It's a combination of a lot of things, right? And also, on the other hand, there is a lot of things going on, to lead to that endpoint or to lead to that status. So trying to use one single surrogate marker in mental health research to study endpoint can be quite challenging, and I totally agree with you on that. Yeah, but if we, let's say, if we do have some markers we're interested, especially for long-term studies. If we do want to look at a combination of things and we want to rank them, and those measures can probably still be helpful in terms of providing some comparisons for us to move into, right? Yeah.
Sounds like the start of an R21.
It's a very interesting.
Right,
yeah, it's kind of a very interesting. This kind of idea. It's actually not new. It's a very old. For any epidemiology 101 students trying to learn to do indirect adjustment, and how do we calculate expected given and then how do we assess that? So essentially, how do we do surrogate by-markers? And probably we'll have to dig into the very old ideas of adjustment. And what do we mean by adjustment? What do we mean by expected versus observed? And what kind of scales we want to use? Shall we use difference? Shall we use ratios? And so how do we interpret those things? And it's fascinating to me how old ideas are still very useful in terms of developing new things. Yeah.
Anyone else? Someone said to me a long time ago, when you ask if there are any questions, count to 15 seconds in your head, and it feels like an eternity.
That's one thing I learned from Tom Fleming is that, you know, when after that 15 seconds, you just say 2 words, hearing none, let's move on.
Which we can certainly do. Well, thank you, Ying, very much for your
Thank
time
you,
this
Ryan.
morning.
Thank you, Susan.
Thank you.
Well, I hope people enjoyed this, and this is again our first BIRCH speaker, first seminar, and there should be more coming. And thank you very much again. Thank you, Ying, and thank you everyone for coming.
Thank you.
Bye-bye. Take care, everyone.
Bye.